Okay, so for our lesson part and our notes part today, we need to talk about some biologically important proteins that work with chemical reactions, and these are enzymes. So just as a review, when we actually have a chemical reaction, we have two parts to it. We have the reactants, which are typically on the left-hand side of the what we call a chemical equation, and the arrow is pointing away from them. And then the products are usually on the right-hand side, and the arrow is pointing to them. So for example, I have an example reaction here. CO2 plus H2O with the arrow, you read that as yields H2CO3 or carbonic acid. So in this case, my carbon dioxide and my water would be my reactants, and my carbonic acid, my H2CO3, will be my products. Now, no matter what type of chemical reaction I have, all of them involve energy. Now, basically, chemical reactions have two choices in terms of energy. They can either absorb energy or they can release energy. And this energy can be in several different forms: heat, sound, or even light. Now, in order to survive, organisms have to have a source of external energy that they are bringing in order to carry out their chemical reactions. And the chemical reactions that support life are referred to as our metabolism. So, that's why we eat, and we take the chemical energy from the food that we eat to actually provide the energy we need for our metabolism. So why do we care about reactions, and why do we care about energy? This is all chemistry. Why are we worried about it? Well, remember, this unit, first of all, is about the chemistry of life, so we are concerned with chemistry. And second of all, it has to do with, like I said, some very important macromolecules. This involves something called activation energy. All of the reactions that occur, even the ones that don't occur spontaneously, and what spontaneously means is basically they occur all by themselves, require energy. So the energy needed to get the reaction started is what we refer to as activation energy. And I kind of think of it as the little push of energy that we need in order to start a chemical reaction. Think of a ball sitting at the top of a hill. It's just going to sit there unless you give it a little push. That little push gives the ball energy. That's the same thing that is happening here with the activation energy. Now, this one here, I'm less concerned that you would actually recognize this. I'm more concerned that you see on both types of reactions, the reaction that absorbs and the reaction that releases, on both of these we actually have marked an activation energy. So it doesn't matter what type of reaction we have, and there's only two choices. We have to have that activation energy that is present. All right, so again, how does this all tie into biology? Well, this is related through the use of enzymes. Enzymes are special proteins that act as catalysts. Basically, a catalyst is something that speeds up a reaction without getting used up itself. So what enzymes do is they actually work inside of living cells, and they speed up the reaction inside of our living cells. And how they do that is by lowering the activation energy. So you actually have some space in your notes, and what I want you to do is actually make this sketch. Okay? What we're going to be doing is we're going to be labeling this. So please pause this as you need to so that you can write everything. Now, what you have to understand is the beginning part here where the two lines meet are called the reactants. Mine's written a little big, but that's so that you can see it. And then at the end where they come back together, that represents the energy level of the products. Now, this top line is the reaction pathway without the enzyme. So this is the energy path that this reaction would take if we didn't have an enzyme present. And this larger arrow would represent the activation energy without the enzyme. So you can see we use a fair amount of energy for that. You can think of it as if you're starting a walk and you're starting at reactants and you're ending up at products, and you walk all the way up a hill and all the way down the hill. You get to the end, and it's going to take you a little bit of time, and you're going to exert a certain amount of energy to climb that hill. By comparison, the black line underneath is the reaction pathway without the enzyme. So notice that the activation energy is represented by that smaller arrow. So let's suppose we took that same walk, but instead of going all the way up to the top of the hill, there was a tunnel or a shortcut that actually took us through the hill instead. We would get to the exact same spot, but two things would happen. Number one, we would get there faster, and number two, we would use less energy because we wouldn't walk as far up the hill. That's exactly how enzymes work. And so what ends up happening is, in this case, it looks like we save about half of that activation energy. This activation energy savings that we get is actually what allows us to survive. Without these energy savings, we would actually not save enough energy for us to continue living. Now, in terms of enzymes themselves, they are very specific for the chemical reaction they interact with. So they have to match a specific shape, and we're going to talk about that here in a second. Enzymes are also reusable, so they can get used over and over and over again in that same reaction before they actually wear out and get broken down. Now, with this being specific, we talk about something called the lock and key model. And in order to talk about the lock and key model, we need to define a word, and that is substrate. Basically, when we use the term substrate, all we are doing is referring to the reactants in a reaction that has an enzyme involved. So it's an enzyme catalyzed reaction. Now, the way this lock and key model works is the shape of the enzyme fits with the shape of the substrate, just like a lock and a key. So I have a key on my lanyard that opens my door and any of the science room doors, but it won't start my car, it won't get me into my house if it is locked. It is specific. Sometimes you'll have a key that actually will slide into the lock, but it won't turn and unlock the lock. So the key is somewhat specific, but it's not specific enough. Enzymes have to be just that specific. The shape of the enzyme and the role of the enzyme is determined by the amino acids and the folding and arrangement of that particular protein, because remember that these are proteins. Where the enzyme and the substrate fit together is called the active site, and that is actually what will, that interaction is what causes the reaction to speed up. So let's look at a very simple geometry example here. So let's say I have my substrate, which is my reactant, and it has a certain shape. I also have my enzyme, which also has a certain shape. You can see that the shape of my enzyme and the shape of my substrate will fit together. And when they come together and they actually meet, we form the enzyme-substrate complex. And the meeting point is what we refer to as the active site. So this enzyme comes in, and in this case it's actually splitting apart our substrate, so it makes it happen faster and with less energy. But at the end I still get the same products, and I get the enzyme that is just the same as it was before, and it can go back and do the exact same thing again. Now, in our bodies or in living things, different things will actually affect the action of enzymes. The main two that we are concerned with are temperature and pH. For all organisms, there is an optimal, in other words, best pH and temperature. Now, in terms of temperature for humans, especially for us, this is 37 degrees Celsius, which on the Fahrenheit scale would be 98.6, our optimum body temperature, and a pH of 7, which is neutral. If we are above our optimal temperature, what happens is the active site will actually denature or deform. And I pull this one out specifically because it's just like if I took my key, again, that was on my lanyard, and I heated it up. Eventually it's going to melt. I can't use it then in my lock. That's exactly what's going to happen. With our pH levels, the exact same thing will happen. The active site will also denature. It's just not melting with heat. It's because of a chemical change that's actually taking place. Now, you can actually see this on a graph. So if I were to plot either temperature or pH, it would look something like this on a graph. And the point that we would be looking for the best would be right there at the peak. So if you were ever presented with a graph saying what is the best temperature for this enzyme or what is the best pH for the enzyme, look for the peak and then find where that peak meets down at the bottom, and that is your answer. Some other factors that affect enzyme action are the amount of enzyme or the amount of substrate. what happens is the concentration of one increases, the activity will also increase to a certain point. So if I increase the concentration of either my enzyme or my substrate, the enzyme activity will go up. But at a certain point, it will level off. So we would end up with a graph that looks like this. Okay, so that kind of wraps up our discussion on enzymes. And tomorrow, what we will discuss is actually water.